So what does an ion selective channel look like? To answer the question, the best method is to use X-ray crystallography. X-ray crystallography is a tool that can be used to identify the atomic and the molecular structure of a crystal. And in this method, one obtains a crystal and then use a beam of X-rays to diffract in many specific directions. By measuring the angles and intensity of these diffracted beams, a crystallographer can produce a three-dimensional picture of the density of electrons within the crystal. From this electron density map, then the mean position of the atoms of the crystal can be determined, as well as the chemical bonds, how ordered and how disordered they are, and many other different kinds of information. So this kind of X-ray crystallography work was performed in the bacteria potassium channel, in this case Streptomycin lividans. And the idea here was that potassium channels from bacteria can be much more easily expressed and purified. And also the potassium channels from bacteria has been shown to be similar to eukaryotic potassium channels, forming functional potassium channels in artificial systems. We start to obtain a lot of insights from this kind of work, and shown in this diagram a very simplified view about how these bacterial potassium channels look like. What you can see here is a cross-section of this potassium channel, and what you can see here is that these channels are very simplified. Each subunit has two transparent domains. It's shown here the TM1 and the TM2, and connecting TM1 and TM2 there is this structure that we call the P region, where the selectivity filter is located. Overall, this potassium channel is 45 angstroms long, 1 nanometer wide, and in this configuration has this V-shaped kind of structure. What's very apparently looking in this figure is that the selectivity filter or the P region is very narrow, about 15 angstroms, and preceding the selectivity filter there's this very large cavity, and this very large cavity would be large enough to accommodate potassium channels that are bound to water molecules, while the selectivity filter or the P region is too narrow to accommodate potassium ions that are hydrated. So we think that potassium ions that goes for the selectivity filter lose their hydration water and then regains when these potassium ions exit the selectivity filter. And in this another diagram, what is shown is you have a better idea about these cavities, and you can see this very narrow cavity here that forms the selectivity filter, and this much larger cavity that forms that precedes the selectivity filter, and it's a relatively long internal cavity as well. And in terms of charge, the amino acid that line those different these cavities, what you can see that the large cavity is the amino acids are relatively hydrophobic. So the potassium ions that enter the channel we still retain their water molecules because these potassium ions will not interact strongly with the amino acid line this cavity. And also you can see a large concentration of negatively charged in the entrance of the channel, and we think that these negatively charged are important to raise local potassium concentration at the channel mouth of this in this protein. And here is another view about how this KCSA channel configuration looks like in another type of diagram. And again, you can see the TM1 transmembrane domain 1 connecting with the transmembrane domain 2 by this small alpha helix, and then you have this p region of these channels. What's very interesting here is this array of oxygen atoms here in the selectivity filter that are part of this signature sequence of potassium channel, the TVGYG motif. So what we think is that the potassium ions interact with these 4 or 5 carbonyl oxygens from the signature sequence TVGYG motif in each of the 4 subunits. And only a single potassium ion can fit at the narrowest part of the selectivity filter without their hydration wires. Just a reminder that the carbonyl oxygen we are referring here in the selectivity filter is this oxygen here. So when amino acids couple to each other form peptides, this oxygen from this carbon here is the carbonyl oxygen. So a better view about how the selectivity filter would work is perhaps in this figure. So here is a longitudinal view of the selectivity filter in these KCSA channels, and in the bottom is shown potassium ions with their hydration shells, and you can see that these potassium ions in the large cavity are still bound to water molecules. And then when the potassium ions enter the selectivity filter, these potassium ions then would be able to replace their water molecules with the interaction with the carbonyl oxygens in the selectivity filter. And these potassium ions would fit perfectly well inside of the selectivity filter, so the energetic cost from potassium moving from the cavity to the another side would be relatively small. This is a very elegant design. This structure is very rigid, so it cannot shrink to accommodate smaller sodium ions. So what you can think about is that the sodium ions can only partially interact with the carbonyl oxygens, and therefore the energetic cost for sodium ions to go through the selectivity filter will be very high. So we can think about the signature sequence TVGYG motif being very important to form this very cage structure that emulate the hydration wars for potassium ions. So in summary, you can think about this potassium channels having this structure with 2 transmembrane domains, having this selectivity filter that's very narrow, that would have customized oxygen cage derived from the carbonyl oxygens, and that this customized oxygen cages would allow these potassium ions to share their hydration wires and then move through this selectivity filter with minimal energy cost. KCSA channels is a bacterial potassium channel that was crystallized in their closed configuration, And more recently, the same group, the Rod McKinnon's group, was able to crystallize another bacterial channel called METK potassium channels, shown on the right, in the open configuration. Now you can see in the open configuration, what happens is that mainly the TM2 transmembrane domain is able to rotate and open this large vestibule that allows the potassium channels to gain free access to the selective filter through diffusion. If you compare the KCSA and the METK channel structures, it seems that we are looking at examples of closed and open potassium channels. And so the understanding here is that to open these channels, the inner helices bend at some point halfway across the membrane, as their C-terminus displaced laterally away from the pore by conformational change during the gating. And if you look the amino acid sequence of the channels, you find that this when these channels transition from closed to the open state, the bending happens in this glycine residue of this inner helix or TM2 domain. And this glycine residue is conserved among other types of potassium channels, such as GERK channels, SK channels, BK channels, and so on. So we think that this kind of mechanism may explain how conformational changes of this transmembrane domains is able to open and close these channels to allow the access of the potassium ions to this channel.